Good morning, everyone. I'm Jay. Now it is 9 a.m. So I will start the webinar session. Firstly, thank you everyone for joining us in this webinar. So in this Octet webinar, we will talk about how Octet can empower advancement in COVID-19 therapeutics and vaccine development. Okay, just a short intro about myself. I'm the field application scientist covering Southeast Asia region, Taiwan, and Australia and New Zealand for Octet Bio. Sorry, there is a sound. Let's see who is now muted. Okay, sorry about that. Let me continue. Okay, so in today's presentation, we will talk about how the Octet platform can be utilized to understand the coronavirus host receptor binding, the neutralization antibody and vaccine development workflow, and also how the Octet can be used for detection and quantitation of antibodies against the SARS COVID-2 in samples, plasma and serum samples. And in the final 15 minutes, we will answer the questions that you have, and you'll be muted, so please input your question in the Q&A chat box. Okay, so today I'm pleased to have Dr. Hongshan Lee with us. He is the Marketing Application Manager in Forte Bio. Dr. Lee has many years of experience in the development and optimization of bioassay platform. After obtaining his PhD in 1996, he held various positions such as an application specialist, technical lead, senior principal scientist, program manager in different life science company. And currently, Dr. Lee is focused on the discovery and lead selection, cell line development, and new technology assessment in Forte Bio. So next, I will hand over the presentation to Dr. Lee. Okay, let's give him a few moments to put on his slides. Okay, Hongshan, please take over. Thank you.
Thank you, Jay. Oh, good morning, everyone. Thanks for attending our webinar. My name is Hongshan Lee, Applications Manager of Sartorius Forte Bio. Today's topic is empowering advancements in COVID-19 therapeutics and vaccine development. I'm going to start with a brief introduction of our products and technology. As you may know, Forte Bio is a provider of Octet system and Pioneer systems. The Octet systems are based on the biolayer interferometry, or BLI, and Pioneer systems are the enhanced SPR, which has the new kinetics assay function. Both quantitation and kinetics analysis of the biologic molecule can be done on these systems. As listed to the left are variety Octet systems from low throughput to high throughput with different sensitivities. The top left one is Octet K2 system, which has 2 channels. The one below it is Octet QKE. After that is Octet Red 96E. Both QKE and Red 96E have 8 channels with different sensitivity. QKE is for large molecules only, and the Red 96E can analyze both small and large molecules. The next one below Red 96E is Red 384, which has 16 channels. And the Octet HTX, that's below the Red 384, can analyze 96 samples in the same time. The bottom left one is the Blitz, which is 1 channel, the low throughput instrument. On the right, we can see many applications can be done easily with the dip and read assay format. The advantage of Octets are easy, flexible, and fast. The key benefit is the crude, unpurified samples can be analyzed on the instrument. As I have just mentioned, Octet can be used for quantitation and kinetics characterization. Since it's fast and easy, many scientists have used it for high-throughput screening. The molecule size of detection ranges from 150 Dalton to nanoparticles and cells. The type of the molecules can be small molecules, peptides, DNA or RNA, aptamer, proteins, antibodies, VLP, and viral particles. Even the bacteria and the mammalian cells can be detected successfully on the Octet. So how does the BLI or the biolayer interferometry work? You can see on the left side, there's the one picture of the biosensor. On this biosensor there is a biocompatible surface built already in the biosensor. We call it the reference surface. The pre-coated chemistry, as well as the molecule later can binding to it, that remain in the solution. What happens is when the white light, actually the light that we use the tungsten lamp, when the white light shining from top through the biosensor and goes to reach to the reference surface and also later the binding captured molecule surface, then the lights will be, if there's more binding, the lights will be also reflected back into the instrument from 2 interfaces. The first interface is between the glass fiber and the reference layer, and this is all the same for all the biosensors. The second interface will be anything captured on the biosensor. As molecule binding to the surface of the biosensor, the path length of the reflection, the one that reflecting from the interface between the surface and the solution, that will increase, while that of the reference reflection will remain the same. So as we can see here, the changes in the interference pattern for all wavelengths result in a spectral shift to the top right. You can see this is the shift. That spectral shift we see in the chart, and we are actually essentially measuring this very small change in spectral shifting. And the output is in a nanometer that show to the bottom right, the y-axis. When you see the sensogram of your data, you are seeing the spectral shift in nanometer in real time. So that's the time versus nanometer shift at the bottom right. So the Octet dependent read biosensor, as shown here, the diameter is 600 micron. It consists of the fiber optical embedded into the polypropylene hub with the sensor-specific chemistry at the tip. Since only the end of the tip of the biosensor being dipped into the sample well for testing, that's why the many crude samples, dirty samples, that can be analyzed directly without clarification or purification, this will save tons of time because you don't need to prepare, do anything samples preparation. And because of that, we can detect, we can do the analyze so many dirty samples listed here on bottom right, like sandy soil, pollen, diesel fuel, milk, honey, crude corn extracts, well, even the cell lysate. Here is a list of the ready-to-use biosensor and the assay kits, which covers many common assays. As listed on the top, we have antibody-related biosensor like AHQ, AMQ, protein A, protein G, protein L, anti-human Fc capture, anti-mouse Fc capture, and Fab2G, which is anti-human Fab CH1. For tag protein, we have HIS biosensor, NTA, and GST biosensor. We also provide APS biosensor, which is hydrophobic biosensor, and 4 different strapped evidence biosensor, which include SA, SAX, SAX2, and SSA biosensor. SAX2 is specific designed for GXP application, and SSA is specifically designed for small molecule applications. The SA biosensor is good for general screening applications, and SAX is good for kinetics assays. And we also have the assay kits. We have 3 assay kits available right now. The first one is the protein A detection, residual protein A detection kit. The second one is the 2 host protein assay kit. The third one is GlyS, which is the sialic acid glycan screening kit. Well, by using the biosensor and the instrument, here is the Octet workflow, which will show you how simple it is. Just to give you an example here. This slide shows an automated kinetics characterization. What you need to do is just to load the buffer in the first column. We're using the black microwell acid plate. The capture molecule you can put in the second column and the analyze of interest in the third column. You just need to load the plate on the instrument and click on the start button in the method setup. The samples can be analyzed in parallel, just like this. As you can see, start from baseline, loading, then the baseline to make sure the loaded molecule doesn't come off. Then you can dip into the association, then dissociation, come back to the buffer. So that's how easy it is. The data is displayed in real time, and the KD and on-off rate can be obtained easily. For the high throughput screening, Octet HTX is the 96-well systems which can quantitate 96 samples at the same time in less than 5 minutes. Now let's take a look to see how ForteBio Octet applications can help accelerate the COVID-19 cure or vaccine. Currently, there are 3 common strategies to find the cure includes the repurposed drugs, that's on the left. For example, the investigation of already approved drugs or for other diseases like SARS or MERS to see if they are effective to treat COVID-19. The second one in the middle is biotherapeutics and antibodies. For example, the design of small molecule or peptides that can target virus-host interaction with the virus replication. Then, for example, this SARS-CoV-2 spiked protein and SE2 recognition, that interaction. All the antibody can be isolated from COVID-19 survivors. Then we can also use the animal model that can also be engineered for virus neutralization. The third strategy is vaccine. For example, several strategies to mimic the COVID-19 viral infection to trigger host immune response to generate antibodies against the COVID-19 virus. So Octet has been used to investigate many different types of molecules and for different applications. For example, listed here, it has been used for virus human receptor binding, neutralization antibody and inhibitor development. It has been used to screen and evaluate binding kinetics and affinity of antibodies, to assess epitope diversity and coverage, and to measure titer, vaccine potency, and stability. Today, more than 30 publications on Nature, Science, Cells, and other journals that have used Octet for coronavirus studies since the outbreaks at the beginning of this year. We have listed them on our website. We have the COVID-19 website page, and please check on them. There's more information available there. Next, I'm going to show some selected example of receptor binding, neutralization antibody characterization, cross-reactivity testing, and epitope binding assay using the Octet. The first example is the evaluating CoV-spiked protein binding to human SE2 receptor. In this case, either SE2 receptor or antibodies could be immobilized on the biosensor. Then you just dip into the crude supernatant sample. Then the data showed up here indicates the SARS-CoV-2 has the higher binding affinity compared to SARS-CoV, which was published on the Cells a couple of months ago. Similar to SE2 binding, this is an example of identifying synthesized peptides that binds to SARS-CoV-2 spike protein with very tight nanomolar affinity. This is a great way to validate inhibitors that can block the SARS-CoV-2 and SE2 receptor interaction. This is an example of COVID-19 antibody screening from the available SARS and MERS neutralizing antibodies. The results showed here that CR3022 and 420CD antibodies have nice binding to SARS spike protein. This can be easily validated on Octet by using AHC and SARS spike protein biosensor and the cross-reactivity testing. And this shown up here. Using similar approach, another group has shown that CR3022 and 240CD is binding to a different epitope other than SE2 binding site. The assay is very clear. The results when you show the spectrum is very clear, and the assay is straightforward in this case. Octet can also be used for understanding receptor binding conformation and accessibility. If we look at the left side, the 3D structure of the receptor binding domain CR3022 indicates that which his epitopes is fully accessible when the RBD is in the up conformation. Octet data show here that the robust binding to non-stabilized spike protein, which is shown up on the right in the graph, is in the green on the top of the curve. But the low binding to trimer S2P and the increased CR3022 epitope accessibility upon trypsin treatment of the trimer. When you use the trypsin chopped up, the trimer, it will open up. Then we see from bottom up the binding increase. These results also show epitope accessibility didn't improve by human SE2 binding. So binding results can be quickly obtained to show the relationship of the function and the structure. Epitope information is crucial to identifying or engineering monoclonal antibody that targets a specific function epitope if we combine together with a favorable kinetics and affinity profile. Octet provides all of this functional assay. It has the flexible assay formats. As shown here, we can choose 3 different assay formats. In tandem assay, as shown to the left, is good to screen multimeric antigens. In the middle, the classical sandwich assay can be used under most conditions and using the crude sample. The premix assay to the right is very rapid. You can use that if the samples is the purified samples. We all know that hardest part for binning is the data analysis. Octet provides HT software that can combine data sets from different epitope binning experiments from day to day, week to week. They combine together and can do the analysis and provide a binding blocking profile and the bin chart to show the antibody blocking relationships, which makes the data analysis much easier. In this slide, both CR3022 and 240CD recognize the same epitope within the SARS-CoV-2 receptor binding domain. Octet is a very powerful tool for this type of assay. Inserting the COVID-19 neutralization antibody. One way is to use the hybridoma method as shown in this slide. In the Nature Communication article, they showed 4 out of 51 supernatant candidates tested to show CoV-2 cross-reactivity, and only 1 candidate, it's a 47D11, showed neutralization activity. Once the candidate is selected, Octet was used for further characterization. The data indicated 47D11 binding to S1b of the SARS and the COVID-19 with a similar affinity. Also, the 47D11 binds into CoV S-acto domain with about 15 times fold higher affinity than to CoV-2 S-acto domain. This is an example of panning platform utilized SARS-CoV-2 receptor binding domain and spike S1 as antigen, resulted in the identification of antibodies targeting 5 types of neutralizing or non-neutralizing epitopes on the SARS-CoV-2 receptor binding domain. Data in this slide is about single domain antibodies targeting 5 types of the SARS-CoV-2 epitopes. Are identified quickly on Octet with several antibodies showing promising neutralization efficiency. Here is an Octet workflow in isolating and characterizing neutralizing antibodies from the COVID-19 survivor. Start with the isolated B cells, variable region encoding the heavy and the light chain were amplified, cloned, and transfected. Clones were screened by Octet for antibody binding with CoV-2 receptor binding domain and followed with binding characterization and neutralization. Then blocking assay and epitope binding assay can be performed. In this case, B38 and H4 showed protection against the viral infections in human SCE2 transgenic mice. B38 receptor binding domain binding site were further evaluated. This method was published on Science. Recently, Rebecca Dubois and colleagues at the University of California, Santa Cruz have developed a new Octet assay for the detection of antibody against SARS-CoV-2 in plasma sample. The 20 minutes BLI ISA assay, ISA stands for immune sorbent assay, that use the plasma samples that provide a real-time measurement of both total antibody levels and the specific antibody isotope, which meets or exceeds the performance of the high complexity methods such as ELISA and chemiluminescence immune assay, which showed very good value on this Octet assay. This method can be immediately implanted for use in urgent study, such as monitoring serial conversions in community or evaluating the effectiveness of potential vaccine. This right now is a highly demanded assay. This assay could be also further developed to provide a novel diagnostics platform for the evaluating of patient antibody concentration and other biomolecules in the clinical specimen. Now, this is very good for the patient right now if they need this, because the assay is so fast, less than like the 20 minutes. Let's take a close look on the experiment. This experiment, what they did is start with the biosensor equilibration, only 1 minute, then load the antigen for 2 minutes, then wash to monitor the tight binding of the antigen. The total antibody binding is 10 minutes. Only the high affinity IgG and IgM will be remaining on the sensor after washing. Final step is the 2 minutes detection. The whole process is 18 minutes in a fully automated format. They did validation on this assay. The same patient samples were tested with ELISA, Octet, and also the clinical diagnostics. The comparison data listed here suggests that BLI ISA assay can detect and rank seropositive samples at a single plasma dilution. So the take-home message is this new Octet assay that has improves COVID-19 antibody detection. Octet biolayer interferometry immunosorbent assay utilized a single-use biosensor in a fully automated deep and read format that providing real-time optical measurement of antigen loading, plasma antibody binding, and antibody isotope detection. This method can be immediately implanted for use in urgent study, such as monitoring serial conversions in community or evaluating the effectiveness of potential vaccine, and could be further developed for provide a new diagnostic method for evaluation of patient antibody concentration in the clinical samples. There's so many examples here. This slide is just a snapshot on coronavirus research using Octet. Actually, today, as I mentioned earlier, more than 30 COVID-19 publications are available. So it's also available on our website. Let's switch gear a little bit. In addition to COVID-19 related work, Octet has been also widely used for titer, potency, and stability assay of different types of virus, like influenza virus and VLPs. The main reason that Octet is very popular for quantitation is easy, quick, and low cost because of the regeneration, and can use the crude samples directly. As shown here in this slide, you just need to load the samples into the well and click the Start button. The 96 samples can be analyzed in 15 to 30 minutes using Octet Red 96E, and Octet HTX can read 96 samples in less than 5 minutes. And that's the regeneration right now. Then come back, pick up another new samples, continue. And as shown up here, once the assay is done, the instrument will generate the standard curve for you right away, and the unknown samples that's in the red dot will show up, also report out in your Excel format, Excel sheets. You got them all in less than like 20 minutes. The whole process is less than 20 minutes. As I mentioned in the BLI ISA assay earlier, Octet can replace length conventional ELISA by using either one step or two step or three steps assay as indicated here. Since all steps can be monitored in real time, it makes the assay development pretty easy. You can stop anywhere you see that's good enough. Like if the one step assay is good enough, you just stop there. Many HPLC, the assay, can also be replaced by Octet. This is one example from Biogen IDEC cell line development group. In this case, if we just look at 96 samples and analysis time, Octet was able to reduce the time from 19 hours, that's on HPLC, to 20 minutes on Octet in this case. This is also being published. Excitingly, this year, we have successfully developed a rapid automated AAV virus quantitation assay. The assay workflow indicates over here is very simple. The table to the right shows there's almost no significant matrix effect on the Octet AAV assay. Compare with ddPCR and ELISA, these two are most commonly used today for quantitate of the AAV virus. There's a good match of the results obtained with purified samples. That means these purified samples after enrichment, which showed the yellow bar here. Octet results, if you look at the black bar, there's a slightly difference between the ELISA and the ddPCR compared with Octet, which could be due to the interference of the ELISA ddPCR assay by impurities in the sample. But Octet doesn't have that issue, the matrix issue. So Octet assay has shown that has low matrix effect with perfect good CV. It's less than 10%. We have the app notes available online. Overall, the data indicates better performance of the Octet assay. From the flow chart, we can see here that will tell you how fast is the Octet assay. Octet assay is less than half hour on Octet Red96E, but 4 to 5 hours with ELISA. If you just look to compare these 2 times, you saved 90% of the time. You got a fast time to results to Octet using the Octet HTX AAV capsid titer compared with ELISA. In summary, here is the reason why use the Octet platform for your research. Octet is an all-in-one platform with the very easy to use the software. You can use this Octet for therapeutic investigations, the research, and also for the manufacturing like a bioproduction. Because Octet provides high throughput kinetics interaction analysis solution to select antigenic targets, vaccine, and therapeutic candidates fast. It's the fast and flexible platform for antibody epitope characterization and coverage determination. The efficient workflows with a wide range of biosensors could be tailored for vaccine and different biotherapeutics development research. It provides versatile solutions in upstream and downstream vaccine development and manufacturing, such as vaccine potency, stability, and titer measurement. As I just mentioned, the easy-to-use data analysis software tool is a bonus. That's why today we have over 2,000 instruments installed worldwide, and the users are in pharma, biotech, academic, and the government. We prepared more coronavirus-related resources here, listed here in this slides. By the way, we're going to share these slides with you later after this presentation. For reagents-related resources of the COVID-19 research, please check the link. We provide 2 links here in these slides. Well, that's all my presentation. Thank you. And let me hand over to Weixing and Jay.
Thank you, Hongshan. Thank you for this very informative presentation. So right now we'll move on to the Q&A session. You can either type your questions into the chat box on the right side of the panel. Okay. So I'll give you all some time to type. Okay, there's one question. Hongshan, so the first question is how to immobilize small molecules and detect finding of DNA aptamers. Is this possible to be done on the Octet system?
Yes, there is some publications available for aptamer assay, Octet assay, and also for DNA and RNA binding assay. So just to be aware, the DNA and RNA, the conformation is slightly different from protein, and it's a linear. So the best for the assays development, I would recommend start with SA biosensor, just kind of like a screening your method first. And once that you know which direction to go, we have, as I mentioned, we have 4 different type of the strep evidence biosensor. Now, in this, I would strongly recommend that you, when you synthesize this DNA or RNA, if you have, if you can do it, just add one of the biotin on it. Then you can use our commercial available strep evidence biosensor right away. And for if you have the very short sequence of DNA or RNA, try the SSA biosensor because that has a high capacity of binding. Any more questions? We have.
Okay, so thank you. Next question is, let's see, can the samples be regenerated? And particularly in serum or plasma?
So you, if I understand this question correctly, the question is, can we recover the plasma samples after the assay? So because we use the dip and read, we don't use too much samples. Actually, we only capture like femtomole on the biosensor. The answer is yes. After the assay, you can recover your samples.
Okay, then maybe to take it one more step, can the sensors be regenerated and reused, the biosensors?
Yep, the majority of our biosensor can be regenerated. For example, the protein A, protein L, the AHC, the antibody-related biosensor, they pretty much can be regenerated for kinetics assay and the quantitation. Like a protein A assay, the manufacturing recommended method is guaranteeing regenerate 10 times. That's just one example.
Okay. Okay. Thank you. Let's see. Let's move on. The next question is, what is the best strategy for analyzing FC receptors interaction as they don't fit the 1:1 model?
Oh, that's a good question.
Analyzing FC receptors.
Okay. Yeah, that's a really good question. Actually, we have recently, actually one of the company using the FC captured biosensor for their all the functional assay for the approved for the drug. So if you do this FC related work, So the good news is we have prepared variety of the Fc receptor, the biosensor. We have the human, we have the mouse, we also have Fab2G, that's the CH1 fragments. So depending on what exactly design of your molecule, you can just choose one of them. As I have shown here in this presentation, one approach is using the AHC capture and then dip into the samples directly for this Fc binding.
Okay, I think we can share a bit more on this strategy directly with the user. You can, so if you have more questions or attendees, you can write into us. So next, let me move on to the next question. Okay, so is it possible to measure the binding affinity of antibody to virus-like particles with SARS-CoV-2 spike on the virus-like particle surface? So I think the question is it possible to measure the binding affinity of antibody to the virus-like particles with the...
Yeah, the answer is yes. The principle is just like the one we did the AAV virus. That's whole virus quantitation. So you just need the good antibody on the sensor. You can either using Baltin labeled antibody using the strep eviding biosensor, or you can use like antibody fragments Fc receptor related. Then you take one of our commercial available biosensor. Once you get that capture molecule on the biosensor, then you just dip into the virus. That's the way that you can do the test.
So is there a preference on like whether the antibody should be on the surface or the virus-like particle?
We strongly recommend to start with antibody on the surface on the biosensor, then dip into the virus.
Okay. So as for the recommended method, you were saying is to capture is to use streptavidin, or is there other sensors suitable?
If you have the Baltin labeled reagents, streptavidin is the best choice. So if you have some antibody, you can also try the protein A or protein G, or the AHC. That also works if it's a human. If it's mouse, you can use AMC.
Okay. So thank you. So let's see, we still have a bit of time. So there is another question. Could you please explain the advantage of using the existing method for detecting COVID with the BLI-ISA? So I think this question is trying to ask what's the advantage of just now the method that you mentioned in the BLI-ISA.
Yes, the major advantage for this is fast. It's less than 20 minutes, and the assay is very short. Also, the sensitivity is pretty good. So as demonstrated on the publication. So this method also showed you can just use the one dilution and just the crude plasma samples. One dilution that works very well. So and the results matches the traditional method ELISA and other clinical method. So that means first, it's for the ranking purpose, it's pretty good, and it has the sensitivity, and it has the shortest the time. So just because that, you can just implant it to any urgent situation. So this might be one of the best assay, and for the antibody based the detection. Yeah, that's the advantage.
Yes,
And it's fully automated.
Okay, let's see. Okay, I'm going to ask you two more questions. So, please explain a bit more on the methodology to measure the neutralizing activity of the SARS-CoV-2 antibodies. So it's asking about how does the BLI measure the neutralizing activity of the antibodies? Maybe explain a bit more.
Yep. Basically, the principle is so-called neutralizing is something that binding to the virus, that blocking the virus bind interaction once you have something on it. So these reagents will be we call the neutralizing antibody, because this antibody has a good affinity that compete with the real binding. For example, if the COVID-19 virus that has the spike protein there, that has interaction that needed to getting close to the cell surface, attach the ACE2 receptor. But if we can have something either already binding on the ACE2 receptor, or something the antibody already recognized the spike protein and faster and more tighter than ACE2, and in this case, they don't have the chance to binding to the cell. If they cannot affect the cells, we're safe. So that's the principle. Then we just test the binding or not. Actually, the Octet everything is based on the binding. Hope that answered the question.
Okay, thank you.
You just need to design the assay accordingly, then to make sure you understand the everything happened in there, and you can block this virus binding.
Yes. Okay, thank you. So if one last questions, can custom sensors be manufactured with antibody of choice? Do we provide custom sensors?
Yeah, we used to do that, but please check with your sales manager, local sales manager, because from different time to time, they might change a little bit. As I know we used to have this service. If you have the demand, you need something that's in a large bulk, and you want us to make the kind of like GXP standard biosensor just for your purpose, and that's kind of like our service team will do that. That was before. I don't know recently. I haven't followed that closely, but I can find out get back to you. And also I want to recommend to contact your sales manager, and they know better probably than I do.
Yes. So the email written on the slide now, if you have more questions, you can reach out to us. So right now it's already 10 a.m. If you have more questions, please reach out to us and we will reply you shortly. And thank you everyone for your time with us. and I look forward to seeing you in our next Octet webinar. And that's all. Bye-bye.
Thank you. Bye.